Good morning, ma'am, and good morning, everyone.
due to some ચાવી ખોવાઈ ગઈ છે ચાવી કોકના રોંગ એન્ડમાં નથી પણ ચાવી સાવ ખોવાઈ ગઈ છે આ બે લોકેશન છે અને પછી આપણે મારી સાઇન કરીને દેતો એટલે એ આપણને ડુપ્લિકેટ કી આપી દે બરાબર એટલે લેટર એક સારો ડ્રાફ્ટ કરી દે હવે આવું કન્ડિશનના લીધે ચાવી સાવ ગુમ થઈ ગઈ છે રોંગ એન્ડમાં નથી પડી ગઈ પણ લોસ થઈ ગઈ એટલે કમ્પ્લીટ થઈ ગઈ
So, good morning, everyone. I hope I am visible and audible clearly. Rekha madam? Okay. Oh, yeah. Yeah, okay, okay. Thank you. So, I guess I should start. Okay.
Or you can wait for 2 minutes.
Yeah, sure, ma'am. Yes, ma'am. Please. That would be better. Um, I guess I should start now. Okay. So, good morning, everyone, and thank you for joining for the webinar. So, before starting today's topic, okay, I would like some of students should write an answer in that chat box. Okay. So, I'm requesting all the student participants if they can write the answers, okay, of the first question, just to know, what they think of this. So the first answer I have came, okay, from Tejal Gohil, Hasti. Okay. So that is MDR. Okay, you now know that it is multi-drug resistance. Okay, fine. Now I want every one of you to write name of at least one antibiotic. Okay, at least one name of at least one antibiotic. Okay, that you know, that you know. I want to know some. So Shelja from yeah, azithromycin, erythromycin, chloramphenicol, amoxicillin, penicillin. Ria Satodia has specifically mentioned amoxicillin. That is interesting. Okay, penicillin, penicillium, penicillin. Penicillium is fungi. Penicillin is antibiotic. Tetracycline, vancomycin, streptomycin, ciprofloxacin. Yes, penicillium. Fine, very good. Okay, yeah, doxycycline, 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 doxycycline. Okay, azithromycin again. Good, very good. Now, the next question. Okay, if you have taken any, I mean, if you have mentioned penicillin, ampicillin, gentamicin, okay, are you really sure you have used them? You have taken them in some course of your life? You sure of it? Okay, yes or no? Okay, just those who have said penicillium, ampicillin, gentamicin, if so. Okay, are you really using penicillium, ampicillin, gentamicin now? Okay, no. Okay, so there are some reasons, okay, for which you will not be able to use the antibiotics you have rightly mentioned, or the antibiotics that you already know. Okay, and today I'll try to just brief you, because I cannot take up the complete topic. It is very, very, very big challenge. Okay, today I'll try to touch up or brief of some part of multi-drug resistance, the reason behind you not being able to use antibiotics that you are using right now or you have been using in past. Okay, so I'll go with my topic now. Okay, the multi-drug resistance, I have named it as a next pandemic. Okay, and to be sure, before we can end this seminar today, okay, I will try to prove my point that it is the next upcoming pandemic. It, in fact, it is right now going on, but it is silent. Okay, so first, okay, if we see the total death tolls that till now have encountered this planet, okay, the Black Death, okay, or the bubonic plague that lasted in somewhere about the middle of the 14th century, 1350 around, okay, that caused, okay, eradication of about half of the population of the earth, 200 million, it is said so, and it was worldwide. Okay, I guess you should, if you are microbiologist, okay, or life science student, you should know the causative agent of bubonic plague, and not just bubonic plague, the smallpox, Spanish flu, okay, the plague, the HIV AIDS, which is still now consistently growing. Okay, all these other flus and poxes might have eradicated, Okay, they are no longer existing, but HIV AIDS is still a biggest pandemic endemic. Okay, and there are few more. Now if you can say downstairs I have written coronavirus COVID-19. Okay, do you have, if you are reading newspaper and going with the trend, okay, do you know what is the death toll? Okay, that toll that coronavirus has caused to this earth, this planet. Okay, I just want to know the number. Okay, at least 4 5 students, if they know, can drop a message in chat. I just want to know the number. Around what? Around how many people have died? Okay, because of the coronavirus infection that is existing right now. Four lakh, 360K. Okay. Any more? More than 3 lakh? Is that then okay? 3.5 lakh, 2.4 lakh. Now, let me show you. Okay. The position of coronavirus in this figure is not actually correct. Okay. If I show you the correct image, okay, the star is landing somewhere between 600 to 200. It is actually near about 400K. Okay, which is about 4 lakhs. Okay, and still counting. Still counting. Okay, so it has progressed a lot. Okay, and we don't want that to happen. Now these all are infections may be caused by different type of microorganisms ranging from bacteria to virus. Okay, some may be because of mixed infections also. Okay, and fungal infections or parasitic infections are also one of the causes, leading causes of deaths. Okay, now before going further, okay, there are two terms that I want you to understand: antibiotics and antimicrobials. Okay, antibiotics are small molecules. Okay, they are not large enough. Okay, they are small molecules that are produced by microorganisms to kill other microorganisms. But antimicrobials can be natural, can be synthetic, can be man-made. Okay, they can be compounds and they can be agents like heat. Okay, heat, phenolic compounds. Okay, heavy metals, they can be naturals, they can be man-made. Okay, like bleach. Okay, antibiotic itself is an antimicrobial. Okay, so these are two different words. Sometimes an organism can be antibiotic resistant and sometimes it can be antimicrobial resistant. For example, tolerance to heavy metals. Okay, heavy metal tolerance, heavy metal generally kills microorganisms, but if it is not killing it, then it is antimicrobial resistant. Okay, so these are two basic differences between antimicrobials and antibiotics. You should know about it. Okay, now moving on to further. uh, After the First World War, okay, the discovery race of antibiotics had started. Okay, uh, I guess you know who discovered the first antibiotic and when. Okay, if someone knows, I'll invite them to answer them on chat. Okay, Alexander Fleming, very good, Pathraj Singh. And when was it discovered? The year, if you know. It is already actually depicted in the discovery timeline. Okay, it was at the end of the First World War. Okay, then when it was discovered. Yes. No, Pathraj, you are very late in that. Not 1945. Okay, it was actually in 1920s that antibiotics were being discovered. Okay, although Alexander Fleming discovered it first. Okay, in fact it was not commercialized. He discovered it. Okay, but it was not commercialized. We will see to some of these. For example, these classes, okay, are based on the chemical structure that they have. Sometimes they can also be classified based on their range, whom they are killing. They can also be classified based on who they are, by whom they are produced. They are produced by fungi, they are produced by bacteria, they are produced by soil bacteria, etc. Okay, the first class of antibiotics, beta lactam, then came the then came the sulfonylamides, and sulfonylamides were first commercialized. Okay, it was followed by aminoglycosides, tetracyclines. Okay, macrolides, etc., chloramphenicol. Okay, all these are very big group of very big classes of antibiotics, and they have their specific applications or ranges. Okay, this was I'm talking about. Okay, when they came in market, when they came in the commercial use. Okay, if you see this, okay, in so sulfonylamides came to the market in 1936. That was before or before the penicillium. Okay, although it took almost 20 years for the commercialization and application of these, and to find the targets whom they are killing against the pathogens. Okay, and since then there are several discoveries. You can see 2013. Okay, in fact there are few more. This taxibactin, the last one. Okay, it is being investigated for several applications. Okay, and it takes time. Okay, now I'll just quickly go through it. I won't go in deep of it. Okay, the biggest class of antibiotics that is prevalent, okay, are beta lactams, and it is not just the only one. Okay, penicillium, amoxicillin, fluoxetine, and there are many more. Okay, cenflexins, they can inhibit the bacteria based on the some modifications, like for aminoglycosides. Okay, they inhibit the protein synthesis that kills the bacteria. The chloramphenicol, okay, it was the first line of defense, the first drug that was given, but it's no longer accepted now. It's no longer given. Okay, sulfonylamides, the first commercial antibiotics, no longer given. They have restricted uses. Okay, they do not kill the bacteria, but they stop their multiplication. Okay, but they also cause allergic reactions because sulfonylamide compounds can cause trouble in your metabolism. The tetracycline. Okay, there was a time when it was again a first line of defense for veterinary doctors, but now no longer. Okay, and there are glycoproteins, there are others, lipopeptides, streptogramins. Okay, there are many more. Okay, you can get this image from Google, and in the end of the presentation I'll share some links which will be very helpful to you if you are interested to study the mode of action of antibiotics, the mode of action of resistance, and etc. Okay, now the mode of action of antibiotics can be depicted in different ways. I have three different images to show that. Okay, basically all these antibiotics actually stop one or more very important part of metabolism of a bacteria or a microorganism. It can stop or inhibit the cell wall synthesis. It can stop the action of DNA gyrase enzyme. Okay, it can stop or inhibit the action of DNA dependent RNA polymerase. It can stop the ribosomal activity or RNA synthesis. Okay, it can inhibit the nucleotide synthesis, the folate synthesis inhibition actually. Okay, the para amino benzoic acid, paba, then DHF, then folic acid. This folic acid again is required for nucleotide synthesis. If nucleotides are not synthesized, bacteria will die. It will die. Okay, now some of these action are bactericidal or static, means either killing the bacteria or stopping the growth of bacteria. Ultimately the aim ends in stopping the infection. Okay, now the same thing can be depicted in different ways. I have depicted the same thing. Okay, and here you can see that cell wall synthesis inhibitor list is followed by the antibiotics that are being used. Okay, penicillium will stop the cell wall synthesis. Its near relatives, the vancomycin, bacitracin, etc. Okay, sulfonylamides will stop the folic acid metabolism. Erythromycin, okay, the clindamycin, okay, chloramphenicol, they stop the protein synthesis. Now they have their side effects also. I'll tell one side effect. For example, chloramphenicol. If you give chloramphenicol, it will inhibit the 50s subunit of the ribosome. Okay, but this 50s subunit is also present in our mitochondria. So our mitochondria will also get affected because it is of bacterial origin. It has the same ribosomal machinery. So chloramphenicol can inhibit with the mitochondrial action, and this can cause side effects. And it's not just about chloramphenicol. These many of these antibiotics do have side effects. Okay, if they are not taken judiciously. Is there any question? I guess I'm seeing in chat. Okay, yeah. So it was 1928. Okay, so I'll continue this. Again, different way of presentation of antibiotics and their mode of action. Okay, how they are working and what they are working on. Okay, so essentially if you stop one or more of any key metabolic step or key regulatory step of the growth of a bacteria, you can kill it. Okay, and these antibiotics are doing the same thing. Okay, they are interacting with the biomolecules of the bacteria, which are very important for it. Okay, and hence stopping their growth. Now uh, the data shows, this data was published in a very good journal, PNAS. Okay, Proceedings of Natural Academy of Sciences, and it was also broadcasted in the Indian newspaper Hindustan Times. Okay, so the data shows that India is the largest consumer of antibiotics. Okay, and this consumption has doubled in 15 years, in last 15 years. Okay, you can see that it has ranged from 3.2 to 6.5. That is again, these numbers are not, they are in millions. Okay, so India is ranking, although it was already ranking first in antibiotic consumption, but in last 15 years, okay, and till 2015, okay, it has been shown that antibiotic consumption has doubled in India. But this doubling of the antibiotic consumption has not decreased the infections, but it has added, it has increased the number of common infections like E. coli, streptococcal throat infections, pneumonia, and tuberculosis. Okay, so taking more amount of antibiotics won't help. Okay, but it is a different aspect. Now, this is not the recent data, because whenever there is any medical data to be shown, they are actually counted and calculated every 5 years, like our census. Okay, they are counted and they are evaluated and analyzed and then presented. So the data that we have may be of 2015, 2017, because the data of 2019 and 20 are still being calculated, and they will be available to us in maybe 2021. Okay, so this scenario is 5 year old. You cannot imagine the scenario currently, and it is very devastating. Okay, let's see the time game. Okay, earlier antibiotics were able to kill or inhibit or stop the growth of bacteria. Okay, or biology students would know that this is how we actually test the antibiotics. Okay, these are the antibiotic discs, and when they are kept on the bacterial growth agar plates, they can stop the growth of bacteria. Okay, it shows that they are susceptible. Bacterial growth was inhibited by most of the antibiotics, but now the same bacteria has evolved against the same antibiotics. This is resistance. Okay, and now the bacteria is hunting the antibiotics. Okay, bacterial growth is not inhibited by antibiotics that were classically used. Okay, few of them are still working, as you can see. Okay, but majority of them are not working or working less effectively as previously. So this is a time game. Okay, and this is really evolving. Now we are in actually a chess game with microorganisms. Okay, there are cycles. Okay, when an infection is caused, we take antibiotics, but if after taking antibiotics the organisms are slightly becoming antibiotic resistant, okay, which is not advisable, we don't want it. It's a counter attack. Okay, microorganisms are attacking us, that's an infection. Okay, to counter that we are taking antibiotics, and now to save their lives from antibiotics, the bacteria are becoming antibiotic resistant. Now what should you do? Okay, it's your turn. Okay, what can we do? That's again the challenge. Now if you are using mobile for your vision, you may see a small image. Okay, but this chart that I have obtained is actually trying to show the infections and their infections and antibiotics that can be used against them. Okay, I'll take a small example of it. I'll zoom it to some level so that you can see it clearly. uh, Incentive care units, which are critically which are used to treat critical patients or terminally ill patients. Okay, these are the range of antibiotics that are actually used against some infections. Okay, and I have starred specifically one. Okay, For example, the Enterobacter faecalis, okay, if there is an infection of that Enterococcus faecalis, okay, you take amoxicillin. If there are streptococcal infections, you can take amoxicillin. But if you have an E. coli gram negative infection, okay, or Moxella infection, okay, or Neisseria, okay, or something like Clostridium and etc., you cannot actually take amoxicillin, but you can actually take it with clavodinate. The amoxicillin and clavodinate increases the range of its of the pathogenic action. Okay, so not all antibiotics can be taken for all infections. Not all infections are treated by all types of antibiotics. Okay, so the range and the spectrum of antibiotics is very important. Okay, and this range is decreasing. This range is decreasing. The antibiotics which were broad spectrum, they were broad spectrum in the sense they could kill many pathogens, are now becoming narrow spectrum. Those pathogens are evolving. Okay, now this is not a friendly situation for us. Okay, now I just had incorporate this image. Okay, uh, microbiologist friends or life science friends would know that bacterial cell wall is actually made up of peptidoglycan. This peptidoglycan will have N-acetyl-glutamic acid and other sugar molecules, okay, with 5 peptide links. Okay, these peptide links are very important. These peptides are actually, these pentaglycines or peptide links are actually cross-linked with each other. This cross linkage makes it a strong bond. Okay, it's a strong wall. Okay, this cross linkage is actually carried is done by an enzyme which is known as PBP, penicillin binding protein or transpeptidase protein. Okay, the main function of this transpeptidase is to carry out the linkage between the new incoming pentaglycines. Okay, and by doing that it will make a longer chain, a stronger chain, and this makes the cell wall strong, bacterial cell wall strong. Okay, and this is because of the action of this enzyme unit, the enzyme known as transpeptidase enzyme, sometimes known as PBP. Okay, now what happens if someone is interacting with this or inhibiting the action of this transpeptidase? The penicillium or the beta lactam antibiotics actually bind with this PBP, penicillin binding protein, and stops its action. They do not allow the transpeptidation. They do not allow the cell wall to be synthesized. They do not allow the cell wall to grow stronger. Okay, like penicillin, cephalosporin, monopenicillin, monobactams, okay, carbapenicillin, etc. These are the antibiotics which are binding and stopping the action of over transpeptidase, bacterial cell wall enzyme transpeptidase. Okay, similarly vancomycin is also inhibiting the cell wall synthesis, but in a different way. In a different way. Okay, you can see the red dots and black cap. Okay, the vancomycin is actually binding to the d-alanine okay moiety of this N-acetylmuramic acid, and if it is binding there, the transpeptidase enzyme cannot work. Okay, now this is the mode of action of antibiotics. This is how antibiotics used to kill bacteria. Okay, but now the scenario has changed. Okay, and sometimes uh, from a mixed population, okay, whenever you take any antibiotic, majority of the organisms or bacteria gets killed because of the mode of action of bacteria of the antibiotic. Okay, but there is at least very small amount or population of bacteria which do not get killed. Okay, they are resistant intrinsically by mutation, by adaptation, or by any chance. Okay, or because of the lack of awareness. Okay, I'll come to that also, a social ethical issue of antibiotic usage. Now what happens, in the first image you can see there is a mixed population of bacteria, the majorly similar type of bacteria. If you take antibiotic, majority of them killed, but at least 0.01% of bacteria survive. Those bacteria after some time do not get killed by antibiotics. They become resistant and their progenies are resistant to antibiotics. Okay, and this property can be shared. Bacteria can share this property with one another. Okay, and this weapon of bacteria, okay, that it is sharing with others is again very dangerous. Okay, now you cannot use your classical antibiotics, although it takes time. Okay, it takes some decades to do that, but a decade is not enough to invent new antibiotics. Okay, so there are key factors or facts which are related with the antibiotic resistance. Okay, so the antibiotic resistance is the biggest threat to the global health, food security and development also. Okay, because this antibiotic resistance does not kill anyone. Okay, but it kills everyone of every age in every country and continent. It is not limited to some country or some part. Okay, and second thing, it is also occurring naturally. Okay, and it is also spread because of the misuse of antibiotics by humans. Okay, now coming to the social ethical issue. Okay, we have a tendency, we particular have a tendency to take medicines or antibiotics only till we get cured. We do not complete the course. For example, I am given 5-day course of some antibiotic because of some infection, stomach infection for say. Okay, I am being told that I should take these medicines, these antibiotics for 5 days. After 3 days I am feeling fine. I am feeling fine. Okay, so I'll stop the usage of the antibiotics for rest 2 days, thinking that I'm already fine, why should I take it? Okay, so now if there are at least one bacteria which is still not killed inside my gut, will develop a resistance towards the antibiotic, and next time if I have some infection, it will not be cured. Okay, stomach infection used to take 1 or 2 days to cure, but now it is taking 3 to 4 days to get cured. Why? Because of antibiotic resistance. The infections that are now coming up are resistant to your antibiotics. They won't get killed. They are growing in number and becoming harder to treat. Okay, now as long as you stay in hospital, you have a larger chance of getting infected. Whenever you go to a hospital, a clinic, okay, or a healthcare facility, you are exposing yourself to antibiotic resistant bacteria. Okay, i'm not I'm not degrading the hospitals, but they are prevailing over there. Okay, and this uh, this is because of their ability. Okay, now we will see their ability. Okay, this is modes of antibiotic resistance, pronounced as ABR. Okay, the first, okay, antibiotic, uh, whenever it is exposed, okay, it should go inside the bacteria and kill it, but antibiotics are not taken up inside the bacteria. Bacteria has modified their import system in a way that the antibiotics cannot enter the cell. If it cannot enter the cell, it cannot kill the cell. This is the first modification that has arisen in bacteria. Second, okay, as soon as it comes inside, throw it outside. It is known as the efflux protein pumps. Okay, there are few proteins which are now expressed at a higher rate in bacteria like pseudomonas. Okay, what will happen? For example, as soon as the antibiotic comes, the efflux protein will throw it outside. So before it can kill the cell, okay, it is thrown out. Okay, that is the second mode of antibiotic resistance. The third, okay, even if by some chance, okay, the antibiotic is coming inside the cell, okay, use an enzyme to inactivate it. Okay, so your drugs or your antibiotics are modified. If it is modified, it will not be able to work. The last and the most lethal ones, okay, alteration of the target. Okay, that means the enzymes which were the attacking site of the antibiotics are now modified. Okay, and the alteration of drug target is supposed to be a development, a evolution, and evolution may always not be good. Sometimes it's bad. Okay, and this evolution is increasing. The bacteria is modifying its enzymatic machinery in a way that it is not killed by drugs or antibiotics. Okay, these four are the main types of antibiotic resistance patterns. Okay, can we close the, okay, Bhautik, okay, nice question, Bhautik. Okay, there are definite ways of doing these things. Okay, Bhautik's question is, what if we can stop the efflux protein? Again, you have to use an antibiotic. Okay, and bacteria are again coming up with the next step. It's a chess game, as I told you, Bhautik. Okay, it's a game of chess. You take one step, bacteria will take another step. You take one step, bacteria will take another step. Okay, next thing, as I told, we are not using it judiciously. We are not taking it in the way that is advised. Okay, you definitely can find a way to stop the efflux protein. Okay, but efflux protein are also present in human body. What will happen if that efflux protein inhibitor is inhibiting your efflux protein? Your kidney will fail, your cells will die. Okay, antibiotics cannot be so harsh that it can kill us. We are taking it in a way that it can kill bacteria only, and only bacteria. Next thing, I'll come with one more example of it. Please remind me, Bhautik, about this when we talk about human microbiome project. Okay, I'll come to this question again, and please remind me of this when we will be talking about human microbiome. I want to show some correlation with that. So these are the four main ways of antibiotic resistance which a bacteria can employ. Bacteria can employ one or more of this system, either inhibit the uptake, efflux protein modification, and target modification. Okay, change the drug or change the enzyme. Any of this can be employed. Now, same thing I have tried to show in different way. I have got the different images from internet. Okay, this is again a diagram showing the inactivation beta lactam ring inactivated by enzymes beta lactamases. Okay, modification of target site. Vancomycin used to attack on D-alanine moiety of N-acetyl acid, right? But now bacteria has modified it. Bacteria has modified the pentapeptide. It has produced D-lactase D-alanine. Now vancomycin cannot bind. It cannot stop the growth of bacteria. Okay, mutation in target. DNA gyrase of bacteria is already modified. Okay, fluoroquinones cannot work. Rifampicillin cannot work on the RNA polymerase. Why? Because the RNA polymerase is now modified. What will you do? Okay, Snehaba, antibiotic resistance plasmid transferred from one bacterial species to another is also responsible for exactly. Okay, Snehaba, very good. As I told that bacteria can share this property. One bacteria, for example, I am a bacteria. I have acquired a property. I have got a property to kill or to inactivate inactivate any antibiotic. If it is plasmid bound, I can share it with my near relative species by conjugation, by transduction, by exogenous uptake. So there are many ways that it can spread. I will come to that also. Okay, so antibiotic resistance is definitely spread by plasmids, and it is largely spread by plasmids, but there are other ways also. Okay, this is a nice question. Now, penicillin, previously it used to act on the cell wall, but now we have a bacteria, we have a different type of penicillin binding protein which is known as penicillin binding protein 2A. Okay, penicillin cannot inhibit it. Efflux proteins. Okay, efflux proteins, it is largely found in pseudomonas species. Okay, these efflux proteins are able to efflux out or throw out tetracyclines, macrolides, fluoroquinones, phenolics, etc. Okay, target site modification, as I said, polymyxin is now not able to work on MR1 fluoroquinones. Why? Because the target is modified. And this modification is not just came in gram positive but also in gram negative bacteria. Okay, bacteria is producing new enzyme, new proteins, new materials, and whatnot, all just to prevent itself from antibiotics that you are using. Okay. Oh, yes, few more. This is the second type, as told. Okay, when it comes to community or biofilm, okay, bacteria has developed the ability to form biofilm, and when the biofilms are made, Okay, these biofilms are very big group of bacteria with different properties. Okay, similar or different bacteria, generally similar. And when biofilms are formed, okay, the antibiotics cannot actually penetrate it. If this is not penetrated, okay, the bacteria inside the cell, inside this biofilm, will be prevented from the mode of action of antibiotics. And biofilm formation is a new weapon for majority of the pathogenic bacteria like pseudomonas, klebsiella, etc., etc., many more. Okay, so biofilm, if the bacteria is forming biofilm, then it is hard that it can be cured by antibiotics because antibiotic cannot enter it. Okay, to give you an example of it, for example, those living in a hostel, can I enter the hostel? No, I cannot enter the hostel because it has limited permeability. Only few people are allowed inside it. Okay, so these biofilms are actually big clusters, big group, big societies, or I can say cities of bacteria. Okay, where many bacteria are living of same type, and they are not allowing the antibiotic to enter. So it is also giving an resistance. Now let's see our heroes. Okay, I should not say heroes, they are devils actually. Okay, these are the top rated MDR pathogens, okay, that are responsible for million deaths in a year in and around the world. Okay, the topping this chart, the first one is the multi-drug resistant tuberculosis. Okay, you can find many literatures about tuberculosis, multi-drug resistant tuberculosis. Tuberculosis used to be a very common non-pathogenic bacteria that was present in hospitals and other things, hospitals and veterinary clinics and what healthcare sectors. Okay, but somehow because of high usage of antibiotics, because of the its conjugation with other pathogenic bacteria, okay, it has acquired genetic resistance towards many drugs. And now this has become a notorious pathogen, although we have found some cure about it, but it still is not able to be completely eradicated from this planet. The second one in this is personally my favorite, MRSA, methicillin resistant Staphylococcus aureus. Okay, third one, vancomycin resistant enterococci. Okay, very pathogenic range of bacteria, gram negative bacteria, enhanced spectrum, beta lactamase producing gram negative bacteria. Enhanced spectrum, it has widened its spectrum. Okay, Klebsiella pneumoniae, carbampicillin is producing gram negative. Carbampicillin is again is an enzyme produced by Klebsiella, and now this enzyme is being spread via plasmid in other gram negative bacteria. Am I getting some question? The characteristics of efflux protein in relation to the concentration gradient or any ionic concentration dependent. uh, i'll try to I'll try to do that when we come, Bhagya, we will try to come on that when we talk about vancomycin resistance, okay, and also pseudomonas. Okay, uh, definitely these efflux proteins are managed by some concentration gradient, okay, and also by the charge inside and outside the bacteria. Oh yeah, majority of these actions are energy dependent. Okay, you definitely need lot of energy to throw out these, to inactivate these antibiotics, and antibiotic resistance is taking great energy from bacteria, but bacteria will do anything to prevent itself. Okay, it can employ energy. Okay, so yes, definitely it is concentration dependent. Okay, it is ion channel dependent. Okay, and it's a membrane potential dependent phenomenon. Now, coming back to MDR, GNR, okay. MDR or GNR collectively stands for multi-drug resistance gram-negative bacteria, and it has been found out that many bacteria which were common residents of our microbiota, okay, like Enterobacter, Klebsiella, E. coli, Acetobacter, Pseudomonas, they were normally present in on the body, soil, etc. Now have become multi-drug resistant. Okay. Lastly, a new group has been formed. It's known as escape. The escape group of organisms are few most notorious pathogens, gram negative and gram positive both, which are affecting not only humans but animals, okay, livestock and agriculture, okay, and they have developed many type of antibiotic resistance. And the escape group actually is formed of mainly Enterobacter faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter, Pseudomonas, and Enterobacter species. Okay, they are responsible for many types of infection: lung infection, stomach infection, skin infection, throat infection. Okay, multi-organ failures, etc. So these are the major types of MDR pathogens. Okay, in fact, the group of pathogens. Okay, this is not just about one. Now I'll try to show 2 3 main types of these pathogens. Okay, and I'll start with Klebsiella pneumonia. The Klebsiella pneumoniae is famous all around the world for causing pneumonia in humans and other animals. Okay, pneumonia is actually a respiratory infection. Okay, it infects lungs, trachea, and bronchi, especially. But Klebsiella is also able to infect, okay, or cause infections in wounds, surgical sites, urinary tract, bile duct, etc. Okay, and other than that, now as it has become a notorious pathogen, a multi-drug resistant, it is also able to infect the urinary tract, upper respiratory tract, cause diarrhea, okay, cause inflammation in vein, gallbladder, also meningitis, brain infections, inflammation in brain's protective layer. It can also go or penetrate till bones. Okay, it can cause bacteremia, that is blood infections. It can cause sepsis. Okay, now any of these situation can be lethal. Okay, and it is said that it is the second or third most common pathogen that is found in urinary tract. Okay, now there are few things that can be known about Klebsiella and where they are very interesting. Okay, that most common cause of hospital acquired patients. Okay, whenever you go to any hospital and you get infected by anything, then most commonly it can be Klebsiella pneumonia, because it is prevalent over there. Okay, all those babies which are premature, okay, when they are born, they are kept in premature intensive care units. Okay, and those babies are more likely to get infections, and this infection is because or can be because of Klebsiella pneumonia. This Klebsiella pneumonia again is the second most causative agent for bacteremia, okay, and urinary tract infection. Now, do anyone of you, do you know which is the first main causative agent, or which is the first most encountered agent in urinary tract infections? Do you know, whenever you have a urinary tract infection, which organism or bacteria will be the most common in it? If Klebsiella pneumonia is second, who ranks first? You can reply me on chat. I mean, hello, am I audible? E. coli, yes. Very good, Bhagya. E. coli is the most common one and the first to be encountered whenever you have urinary tract infection. In fact, it is called UPEC, uropathogenic E. coli. Okay, again Enterobacter, again antibiotic resistant and not good. So Klebsiella is the second one. Okay, and antibiotic resistance, when it was found, okay, it was found that Klebsiella already had many antibiotic resistance, and the first antibiotic resistance new genes, the genes of antibiotic resistance were first discovered in it, and all the types of antibiotic resistance genes were first discovered in Klebsiella. Okay, the timeline below shows genes and time. Okay, in about 1972 SHV1 was discovered. Okay, then TEM1, SHV2, cry genes, KPC. Okay, and these are the names of the genes which are responsible for the resistance towards ampicillin, fluoroquinones, carbamazepines, etc. Okay, so this is a notorious pathogen, and intrinsically by its own inheritance, by its chromosomal phenomenons or genes that are present in chromosomes, they are already resistant to penicillin specific antibiotics like ampicillin. They won't act on it because it has different type of transpeptidase, and those transpeptidase are not inhibited by ampicillin. So intrinsically it is this. And Klebsiella is carrying many plasmids, and it can spread the plasmid in many species, in nearby any species. Okay. Now, scientists had tried to figure out how this common Klebsiella has become so virulent. Okay. And they could find, or they can make a chart of it. Okay. This is a chart that shows that prevalence. Okay. How different environment, animal and human factors can cause Klebsiella to acquire different traits and characters. Okay, let's start with human. Human has Klebsiella in its in gut, in urinary tract, and it was common. But as it came in sewage, it also came with animal effluents. Okay, they all mixed up in the environment. Okay, in the soil and water. Okay, and they shared some plasmid with one another. Okay, with other Klebsiella and other species. Okay, this is known as horizontal gene transfer. Okay, for the undergraduate students it may be a new term, but majority of the master degree student, okay, they know it, what is horizontal gene transfer. You will learn it. Okay, it is giving and taking of plasmids. Okay, this has resulted in evolution of Klebsiella, and in every cycle Klebsiella is taking up new genes and becoming AMR, antimicrobial resistant. Okay, now uh, unjudicious, uncontrolled use of antibiotics in livestock, giving animals antibiotics just so that they became fit and fine. Okay, it has also resulted in increased cases of meningitis and infections in animals, and these animals are again used by humans for feed and fodder. Okay, and for cultivation and other purposes and practices. And if you encounter animals, you come in contact with them or you consume them. Okay, you can get affected, animal can get affected. Okay, and slowly Klebsiella is evolving. It is giving and taking up new genes from one other place and has acquired resistance to many different antibiotics. Okay, now if you are able to see this chart is able to show different type of Klebsiella pneumoniae resistant to several different types of antibiotics. Okay, particularly in India. Okay, if you see a blue color. Okay, it is sporadic reports and it is shown to have oxycycline resistance. Okay, for example in America, okay, it is showing that it is endemic and predominant. Okay, it is resistant to penicillin beta lactams as well as cephalosporins. Now, carbapenem was thought to be a very good antibiotic, but now the Klebsiella, which was used against Klebsiella, now India is a leading center of carbapenem-resistant Klebsiella pneumonia, which was previously a very good antibiotic, but Klebsiella has become resistant to that also, and predominantly in India and in Balkans. Balkans is the southern part of the Europe. Okay, these two, whenever if you count 100 Klebsiella, okay, 50 to 60% of the Klebsiella will be campicillin resistant in India. Okay, and this chart, this world map is showing a color gradient. Okay, uh, there are few data which are not present over there, and this was again reported by World Health Organization. Okay, few countries are safe, few countries are not safe, but India is again gaining development in Klebsiella pneumonia which are resistant to carbapenem resistant, which was class 3 antibiotic. Okay, carbapenem resistant, the same chart. Okay, in a different format. Okay, about 60% of the Klebsiella that are encountered are carbapenem resistant, and it is not about carbapenem. The antibiotic resistance profile of the Klebsiella that is found in India and in few countries like South Africa, it is shown that the Klebsiella is resistant to antibiotics like amoxicillin, clavodinate, okay, aminoglycoside, carbamazepines, fluoroquinones, etc. Okay, so we need to find something better. Clostridium difficile. Can anyone tell me what Clostridium difficile is responsible for? What disease does it cause? What will happen if you are infected by Clostridium difficile? Can anyone please? Am I audible? Or you are finding out on Google that what is it responsible for? Okay. Yes, so I am audible. Inflammation. See, Bhumi, inflammation is a phenomenon. Inflammation will always happen. Whenever your body finds anything unusual, it will inflame it. May it be an injury, may it be an infection. So inflammation is not actual thing. For example, what I'm asking is Salmonella typhi. What does it cause? Okay, Salmonella typhi causes typhoid. Okay, Clostridium difficile is responsible for what infection, what disease? Okay, definitely it is causing infection in colon. That is because it is causing colon infection, intestinal infections. Okay, it has many different effects. Clostridium are generally anaerobic. It can also cause multi-organ failure. It can cause intrinsic bone infections also. Okay, but majorly it is Clostridium and its other relatives are responsible for stomach infections, colon infections, and organ failures. Okay, now to give an overview of it, okay, this image is responsible is can show you that how many genes does it have, how many antibiotics it is already resistant to. Just look at to the chart: cephalosporin, chloramphenicol, ciprofloxacin, ofloxacin, gentifloxacin, tetracycline, vancomycin. Okay, majority of the antibiotics. Okay, what are class 3? Okay, streptogrammin B, this bacteria is already resistant to it. It can throw it out, it can modify its target, it can inactivate the antibiotics. Beta lactam interacting genes are already there, beta lactamases are there, which will not allow ciprofloxacin and other antibiotics to work. Vancomycin resistance is there. Okay, so how do you think you will able to treat this Clostridium infection if it is acquiring few more antibiotic resistances? Okay, and main thing, it is able to form biofilm. If it is able to form biofilm, vancomycin resistance is gone. Okay, metronidazole. Okay, its infection cannot be you cannot treat that by that. Okay, because it is spamming biofilms. Okay, so this is one good example of many multi-drug resistance. Okay, but my main target about today will be MRSA. Okay, the second main target. Now MRSA is responsible for major uh, healthcare acquired infections. It is also called as nosocomial infections. Nosocomial infections are those infections which you acquire from hospital linked or healthcare unit linked infections. Okay, now estimated that to only in USA. Okay, 20 to 40,000 people in USA alone die because of MRSA infections, because their immunity is low, because they are ill, because they are hospitalized. Okay, it is responsible for 20 to 40,000 of deaths per year. Okay, and that is not a small number. Now if you see methicillin resistant Staphylococcus aureus, okay, those who know Staphylococcus aureus very well in the structure and shape, they can correlate the image. Okay, this has been classified into 3 different types. Okay, healthcare associated MRSA, HAMSA, community associated MRSA. Community associated means the infections that you get from simple healthcare clinics or community centers. And the third is the livestock associated MRSA. That is the methicillin resistant S. aureus that is acquired from animals, from livestock like cow, pigs, chicken, birds, etc. Okay, now when it comes to human, it becomes more devastating and more infectious, because this Staphylococcus aureus, which was a normal flora of human body present on skin, upper respiratory tract, etc., it was usually harmless. Okay, but now it is causing serious infection, bloodstream infections, pneumonia, surgical site infections, and over a span of time, with the lapse of time, the Staphylococcus aureus has acquired antibiotic resistance and more virulent factors. Okay, if you are easy to see this, you can see, but otherwise you can close your eyes for some time. Okay, these are very simple images of simple skin infections. I'm still saying simple skin infections. These are just pimples, spider bites and boils. Okay, they are filled with pus, sepsis. Okay, and sometimes people can also find that they are affected by fever and other rashes. Okay, this is because of the colonization of skin with methicillin resistant S. aureus, that too on skin. Now can you imagine what will happen if it is inside your body, inside your lungs, inside respiratory tract? Okay, the same thing will happen inside the body: sepsis, pus, pneumonia. Okay, so skin infections are really common. We generally don't look into it that much, but they can be because of methicillin resistant S. aureus. And if you have methicillin resistant S. aureus on your skin, today or tomorrow it can enter in your lungs, in your urinary tract, in your body, in your trachea, etc. Okay, now why this methicillin resistant is so harmful? What is its secret weapon? Okay, so as we had discussed in previously, penicillin binding protein, which is responsible for transpeptidization and cell wall synthesis. Okay, methicillin resistant S. aureus MRSA has different type of this transpeptidase, which is known as PBPS2A. Okay, penicillin binding protein 2A. It has modified site. Okay, beta lactam antibiotics cannot bind on this penicillin binding protein, and hence transpeptidation cannot be stopped. Cell wall synthesis goes on, and your beta lactam cannot inhibit the cell wall synthesis of MRSA. Okay, this is one thing. There are many more. World Health Organization in 27th Feb 2017, that is 3 years ago, okay, has put MRSA on the priority list. Priority list of the organisms. Some questions? Okay, Haron says that I'm not audible clearly. Can and others please revert me if I'm audible clearly? Am I audible clearly? Okay, so I'm audible clearly. Haron, I'll request you to please check your internet connections, because that can be because of that. Okay, now I'll continue with my slide. So World Health Organization has put methicillin resistant Asorian in its top priority list. Even World Health Organization finds that we should find a cure for MRSA because of its pathogenicity, because of its strengths. Okay, not just because of the penicillin binding protein 2A, but it has few more pathogenic factors. It has cytotoxins, pore forming toxins, which are known as PVLs. Okay, it has phenol soluble modulins, which are small proteins actually, but which are toxic proteins. It has enterotoxins. All in all, these virulence factors or proteins or biomolecules are responsible for killing the cells. It can kill human cells wherever it is infecting because of phenol soluble modulins, pore forming proteins. Okay, pore forming proteins will puncture your cell. Okay, and if it is punctured, everything inside the cytoplasm will come out. It will ooze out, and bacteria will utilize it and will grow, and this will result in sepsis, pus formation, as we have seen in the last image. Okay, let me come to that. This is this pus formation, the lysis of the cells, the deadening of the tissue is just because it has many virulence factors that I named is PBLs, porins, endotoxins, etc. Okay, and the pus and sepsis formation is happening, and imagine this is on the outer surface of the body. What will happen if it is inside the body? It can be very devastating. Okay, now the world chart of methicillin resistant S. aureus, okay, is showing it's prevalent in second world countries and in developing countries also. Okay, America is said to have highest number of increasing cases. Okay, it is also present in India, China, Australia, Southern America. okay, and few more countries, and the data is lacking, okay. And worldwide it is estimated that 2 billion people carry some or the other type of S aureus, which is MDR, okay, and this number is 53 million, okay. I guess this is more than the number. It is increasing with the increasing cases of COVID. It is comparable to that, and it is increasing now. Uh, the Staphylococcus aureus is not only resistant to methicillin, but it is also resistant to many several other antibiotics like fluoroquinones, oxycycline, aminoglycosides, vancomycin, not vancomycin actually, okay, but rifampicillin, okay. So you cannot actually employ these, okay, but particularly in India and in United States, the scenario is oxycycline and microlides, which are the most prescribed drugs. It has become resistant to that. Okay, in India, it has become resistant to fluoroquinones also. It is resistant to aminoglycosides also. Okay, so there are different types of bacteria of same class, but which are more virulent. Okay, now I was about to talk about vancomycin susceptibility E. coli. Someone had a question, and I had asked Bhautik to remind me of it. Can we come up with that question? Bhautik, what were we talking about? And I told you to remind me of something. Okay, sorry, I just remember. I told Bhautik to remind me about efflux proteins, and that is different thing. Okay, now I will come to that also. I'll try to show you that. Now, vancomycin resistant enterococci. Enterococcal infections, gram-positive infections are common. Stomach infections, lung infections, etc. Okay, sometimes skin and nose, ear infections. Okay, but uh, the vancomycin actually binds to the cap of the D-alanine D-alanine moiety. Okay, and thus it can inhibit the cell wall synthesis. But now what has happened is enterobacters have modified the target site. Okay, it does not make d-alanine d-alanine, but it is making different type of cell wall material. So the vancomycin is not able to bind it. If this is not able to bind it, vancomycin cannot act, and enterococcal infections are not easy to cure now. Okay, this is one type of mechanism. Although this is one type of mechanism, but it is very prevalent. Okay, now mode of action of beta lactam. I am coming back to the point. We are again going backwards, but there is a reason. Now, the cell wall precursor, as we were talking about d-alanine, d-alanine, as you can see in the center image. Okay, the structure of this cell wall precursor is much similar to beta lactams like penicillin, carbampicillin, monobactam, cephalosporins. Okay, because of the structural similarity of beta lactam with this d-alanine d-alanine precursor, the penicilliums are able to inhibit the cell wall synthesis because of competition. Okay, it can stop transpeptidase. Now, penicillin resistance. Okay, bacteria now have found out a enzyme. They actually not have found out, it was already there. The beta lactamase enzyme is responsible for breaking the penicillin to panicololic acid. If it is converted from penicillin to palicololic acid, it will not be able to stop the inhibition. So if bacteria is producing beta lactamase, okay, your penicillins or beta lactam antibiotics will not work. Okay, so what can you do? Okay, this is again a counter attack by bacteria. You were using antibiotic, and this enzyme is actually in altering the antibiotic. So the scientists found out another way. Okay, to inhibit the action of beta lactamase enzyme, we found some compounds like clavulanic acid, sublactams. Okay, these compounds can actually bind with the beta lactamase enzyme, can stop it so that the antibiotic can work. Okay, I hope you are understanding clearly what I am trying to say. Penicillin can only work if you apply it along with clavodinate, because clavodinate will stop beta lactamase enzyme. If beta lactamase enzyme is inhibited, the beta lactam antibiotic can work and destroy the bacteria. Okay, and clavulanic acid is a very promising molecule that came up in this. Okay, so clavulanic acid was found out in 1973 from streptococcus, streptomyces species. Okay, it was found that it is a type of beta lactam itself, but it is not actually an antibiotic. Okay, so clavulanic acid was found to inhibit beta lactamase enzyme. If beta lactamase enzyme is inhibited, bacteria can be killed. Okay, and it was found by Beecham's. Beecham's is an organization, company, and you know it. Beecham's later came to be SmithKline Beecham's, and now it is GlaxoSmithKline Beecham's Consumer Healthcare Limited. Okay, and this is a last hope. It was said to be the last hope of. Uh, Sorry, there is a question. If anyone has resistant bacteria infections and other people still power can kill that bacteria, plasma therapy at that time. Bhumi, uh, we will come to it. Okay, please post this question. Okay, again this question in the discussion session. I have this question in my mind and I wanted to ask you. Post it publicly, not privately. Okay, you can post it publicly in this group chat and we will come to it while discussing what can be done in future aspects. Okay, that's a very good question. Now, augmentin. Has anyone taken this and this prescription? Can anyone say? Has anyone of you have ever taken this augmentin antibiotic that is a combination of amoxicillin and clavulinate? Okay, again this is the original product, as the last line says, original research product of SmithKline Beecham's Consumer Healthcare Limited, London. Okay, when they found out clavulinate, they could find a cure to beta lactamase inhibition. Okay, and since that time still today it is used against ear infections, sinus, skin infection, urinary tract infection, pneumonia, respiratory infections, stomach infections, etc. Okay, if you can remember, has anyone taken this antibiotic any time? If not, you are likely to suppose you may find it. You may find it. Now the price of this antibiotic, this combination, is 340 rupees. And just because it is a combination, it's a unique combination of amoxicillin and clavulinate, it was a patented product, but now the patent has ended. Okay, so it is available at a cheaper price. 6 tablets in 300 rupees, 50 rupees per tablet. And if you take this tablet, okay, definitely you can get cure of any infections that are beta lactamase resistant. Okay, because clavulinate will stop the beta lactamase enzyme. Okay, and amoxicillin will kill the bacteria. This was supposed to be the last hope, but now again we are smart and bacteria are smarter. Okay, Indian subcontinent has particularly evolved new type of Staphylococcus aureus, which are again amoxicillin clavulinate resistant. Okay, 46% of the total isolates of Staphylococcus aureus are said to be resistant to amoxicillin and clavulinate. So what will you do now? You found out antibiotic, bacteria found out antibiotic killing enzyme. You found out an inhibitor for beta lactamase, and the bacteria again has a way of dealing with that also. Okay, so now I want to know what can you do. Okay, is there any other way? Okay, now what can you do? I mean, staphylococcus is already amoxicillin and clavulinate resistant. What will you take now? Any other way? This is an open question for all. Okay, first you found antibiotic, then they found enzymes. You found enzyme inhibitor, they found again enzyme inhibition. Okay, so staphylococcus aureus is now again very virulent. It has developed its own potential to deal with amoxicillin and clavulanic acid also. Okay, that's why it is said to be a super bug. because this bug, this bacteria, okay, is a superman. Okay, you cannot treat it till now. Okay, it is developing. Okay, so there's some question in chat. We can, maybe we can use suppressors. Yeah, suppressors can be taken, Himakshi. Okay, but there are few problems with the suppressors also. Okay, I'll come to that also. Suppressors are actually not only suppressing the uh, Staphylococcus aureus, but they are causing neurological disorders, neurological problems, and that is the problem. Okay, you cannot just take anything to cure anything. Okay. Okay, Bautik says, if I mutate its gene by laser treatment, okay, nice thing. But Bautik, the infection is already present on your skin. There are million bacteria on your skin, and not only on the skin, it is present on others' skin, on air, inside the lung, in mouth, in food, everywhere. Where will you modify it? Same thing, Hitat. Gene modification. You can modify a gene in your lab, but you cannot modify the every gene present in every bacteria on this planet. Can you? Can you modify every gene of every bacteria? No. You can only modify that is growing inside your petri plate, not others. So genetic engineering would not help. The bacteria is already employing a very good genetic engineering to become resistant. Your genetic engineering is very weak. The genetic engineering of bacteria is superior to it. Okay, now we'll come to this discussion again. Okay, just a minute. So what can be done now? Extended spectrum, okay, enhanced spectrum Salmonella typhi, okay, which is already antibiotic resistant to ampicillin, chloramphenicol, sulfonamides, okay, and third generation ciprofloxacins. Okay, the outbreak is already going since 2016. It lastly occurred in Bangladesh, in South India, in Latin America, okay, in Pakistan. Okay, the outbreak is still going on. The Salmonella typhi that is now found out, okay, is already resistant to very large number of drugs. Okay, and if we are thinking that evolution is now, this drug resistance has happened just now, no, you are wrong. Okay, so a few researchers found some graves of soldiers who died in World War One. Okay, and from a grave of a soldier who died out of dysentery, stomach infection, dysentery, loose motion. Okay, the soldier died out of dysentery in 2015. He was buried. Okay, and the few scientists try to look on what were the infections. Okay, so they dig the grave again and they found out Shigella. Okay, the Shigella was present in the debris of the soldier, the dead soldier, the matrion. Okay, now they found that this Shigella that was actually a superbug. Okay, it was resistant to penicillin and other antibiotics 10 years before the first antibiotic to be discovered. You said 1928. Okay, in 1928, and this is 1915. A decade before the first discovery of antibiotic, the bacteria were already resistant to antibiotic. Now how this is possible? Okay, how this can be possible? Because antibiotics that you found is right now, you discovered it today, but antibiotics are already there in environment, in nature, since centuries, since decades, since evolution. Okay, bacteria are fighting with each other, microorganisms were fighting with each other, producing weapons and counter weapons and defense molecules. So they are already intrinsically resistant. Okay, they are fighting with each other since decades, and you are using antibiotic now. Okay, whenever a new antibiotic is found, bacteria will genetically engineer itself. Your CRISPR-Cas9 is nothing. You are using CRISPR-Cas9 now, but bacteria are using CRISPR-Cas9 since decades. They are using genetic recombination. They are using mobile elements. They are using all sort of transduction, conjugation, all mechanisms of modifying themselves to be more resistant, more dominant. Okay, so bacteria are already resistant. Some bacteria are already resistant to few or more antibiotics, but the problem is the resistance is being transferred to more number of bacteria and at a faster rate because of human interference. Because of human interference. Okay, a small example of this is this potential route of transmission of antibiotic resistance. Okay, this will answer some of the questions that were raised by a few students. Okay, now this chart I have again obtained from World Health Organization's one of the website maintained. Okay, hospital. Okay, whenever you are going and coming to hospital, you are giving some bacteria over there and taking some bacteria from there. You are taking new antibiotics and making yourself resistant to it. Whenever you are going for freshening yourself, okay, you are dumping few bacteria, okay, antibiotic resistant bacteria in your sewer. Again, hospital sewer is the main source of antibiotic resistant bacteria. Okay, because the patients there they are, okay, which are ill, which are pathogen, which are carrying pathogens in their stomach or anything. Okay, whatever that is going inside the sewer, okay, or the sewage lines, it is going in water and land. If it is going in water and land, it is again encountered by humans, by birds, by fishes, by animals, by sea animals. Those sea animals, these seas and prawns, fishes and prawns, are being eaten up by humans. This water is entering the groundwater. From groundwater it is entering to the plants. From plants it is entering to animal system. Those animals are used for feed and fodder by animals, by humans, again getting new pathogens. The animals that you are using, household animals, are given lot amount of antibiotics just so that they can stay fit and fine, they don't get infections. But this uncontrolled use of antibiotics has increased the number of antibiotic resistant organisms in it. The wild animals in the forest are again new causes. Okay, they themselves have their new microbiota, their own microbiota. The human interference in wildlife, okay, has caused some infections. You know, coronavirus. Okay, the coronavirus has came from bats, but not just bat, but it is also from another reptiles. It is a mixed conjugation. Okay, humans have started interfering in animal system and getting new viruses and new bacteria. Okay, so humans are surrounded by or encountering new antibiotic resistant pathogens. Okay, pathogens are developing, okay, from several different routes, several different ways. Okay, and to add to this, okay, this is one of my concluding slides. Previously we used to think that bacteria live inside us. Okay, bacteria used to live inside us, but the fact is we live inside the world of bacteria. Okay, write 5 and then write 30 zeros. That is 5 million trillion trillion. Okay, 5 million trillion trillion, that much is the number of bacteria that is found by University of Georgia. 5 million trillion trillion is the number of bacteria that are present on the earth. And what is the human population? Few billion, few trillion. What is that? Nothing. Bacteria are more. We are surrounded by bacteria. Okay, if all these bacteria, okay, become resistant to antibiotics which we are using unjudiciously, uncontrollably, okay, they will start hunting us. And what will happen if they all start hunting us? You will have n number of pathogens and you will die in one day. One day. The infection will not carry 3 days or 5 days or 1 week. You will die out of one day. Okay, these pathogens, these bacteria are still commensal, they are not harming us. Okay, but Klebsiella was not harming us few years ago, few decades ago. Salmonella, sorry, it's not Salmonella, Enterobacters, Klebsiella, Staphylococcus, they were all common bacteria. Mycobacterium was also common bacteria, but now they have again gained antibiotic resistance and resultantly they have become pathogenic. What will happen if this 5 million trillion trillion bacteria become antibiotic resistant? Okay, this is the result of human interference. Okay, now to answer your some of the questions. Okay, I'm coming on to the chat. I have received few questions and suggestions from chat. Okay, first thing, Jill, can we use CRISPR-Cas9 system of genome editing? Yes, you can use, Jill. Genome editing, genetic engineering, CRISPR-Cas9, anything you can do, but that will only result in, it will only change the bacteria inside your slide, inside your petri plate, inside your test tube or vial, but that will not change the bacteria that are already there in environment. Can it change everywhere? No, it cannot change. Next thing, genetic engineering. If you have learned genetic engineering, you are using vectors. Okay, plasmid vectors. Those plasmid vectors are carrying antibiotic resistant genes themselves. You are not actually decreasing, but by using genetic engineering you are increasing the plasmids, okay, which are genetic, which are antibiotic resistance. Okay, ultimately what you do, you just throw the antibiotic resistant, after your experiment you throw, discard uh, all your bacteria in sink, in dustbin, or wherever you want. Okay, wherever you want you are discarding it. If you are discarding it, you are again giving new plasmid to environment and creating new pathogens, new antibiotic resistant pathogens. So genetic engineering is out of question. You cannot solve this. You can only use genetic engineering to convert bacteria inside your petri plate, but you cannot use genetic engineering to convert every bacteria in environment. Okay. Now, uh, now Janki has said that organisms have modified themselves for survival. Okay, and animals are higher organisms. Okay, we cannot do so. Janki, whenever it is a small thing, it is easy to change. As it grows bigger, it is hard to change. Okay, for example, your room, you can change your room in one day. Okay, you can keep the things in a way you want, left to right, up to down, whatever you want, but you cannot change the complete system of this building, this college, this university. Can you do it in one day? No, you cannot. Why? Because this is very big. Bacterial gene is smaller. They have mobile genetic and genetic elements, okay, which can be transferred from one place to another. Its genome and its genetic is much different than us. Okay, so if it is different, okay, you cannot change it so easily. The bigger it is, the harder it grows. Okay, you cannot easily convert it. Our immune system, you are saying that immune system modify our immune system. Immune system is a progressive alliance. It develops slowly. Next thing, immune system cannot harm these bacteria. Why? Do you know why? Because these bacteria were common bacteria, they were not harmful bacteria. So our immune system has thought that that these are friendly bacteria, so don't harm them. Okay, so our body cannot identify them as pathogens, but now they have become pathogens. The same bacteria which was a friendly bacteria or normal bacteria, a non-harmful bacteria, has become harmful now, so our body cannot distinguish between them. Okay, bacteria has virulence factors. Virulence factors are those factors which give more strong power, okay, to fight against few things. Okay, uh, so bacteria is again converted its surface. Okay, the surface is modified in a way that it cannot be encountered by our immune system, and that is a problem. Okay, uh, and what was your another question? Okay, Aarti is asking, antibiotics can penetrate biofilm. Aarti, there are few antibacterial agents which are designed in a way, which are being designed in a way so that they can enter the biofilm, but biofilms are designed in a way that it's a protective cover. Okay, so very few antibiotics can enter the biofilm, and that too at a lesser concentration. At a lesser concentration, it can kill the bacteria, but only on the surface. Inside the inside population is very pathogenic. That is a problem. Okay, one more question was there raised by some student. uh, Plasma therapy. Yes, Bhumi was asking about plasma therapy. Now, Bhumi, plasma therapy has its own restricted uses. Okay, plasma therapy can prevent you from one bacteria, one pathogen. one infection. But what we are talking about right now is a broad host range, many bacteria, many antibiotic resistance. What will you do? Humans are not resistant to this. Humans are not strong enough to find so many infections at a time. That's what the problem is. Okay, next thing. uh, do you really think plasma therapy is that easy? No, plasma therapy is not that easy. Plasma therapy requires special medication. In fact, you need to go to hospital for plasma therapy, and if you go to hospital, you are getting again new infection. I'm not saying that you should not go to hospital. Please, please understand that. I'm not saying that you should not go to hospital. I am saying that ill the if the hospitals are not maintained cleanly, if you are not wearing a mask and not maintaining proper hygiene in hospital, you are getting infections. Okay, you may get infections. Okay, the all the medical waste, okay, all the biomedical waste that you are seeing in and around the hospital, even the mask that you are wearing, even the gloves you are wearing, even the medical equipments and everything, whatever it is, it should be dealt with proper way. If it is not dealt with in proper way, it will carry infections, and those infections will be antibiotic resistant. Okay, that's what I'm saying. Now, community centers are the place where many type of people come and go there. They interact with each other openly. Okay, and that can carry the infection from one patient to another, one people to other. Okay, that is again a problem. That's what I'm trying to show or say. Okay, I'm not now challenge.
Mobile number.
Can you please
Challenge in
yourself?
bacteria discover active.
Can you
Drugs.
please mute yourself, Anjali? Thank you. Now I'm here to read, don't worry, I'll read it, Anjali. Okay, now if there are few more questions, we can still found it. But new challenges, if you are thinking that why don't we discover new antibiotics, no one said so. None of you said that new antibiotics, but new antibiotics have their own problems. Okay, public health. What if the antibiotic is treating some disease and causing another infection? Okay, pathogen identification. You, before discovering any new antibiotic, you need to know what it will kill, how it will act, how good it will, route of administration, clearance, half life, barriers, etc., pharmaceutical factors, allergy, toxicity, pregnancy, effect on pregnancy, effect on age. Okay, effect on heart and other diseases. Okay, whether it will be helpful for animals, not useful for animals, etc. So there are many concerns related to new antibiotic discoveries. Moreover, okay, there are few things that can be done. Okay, there are actual practices that are required right now. Right now you need to deal with few things. First,